A very good morning to all and welcome to this webinar for agriculture on gene editing for agriculture, society and sustainable development, prospects and perspectives, being organized by the Tata Institute of Genetics and Society in partnership with Biotech Consortium India Limited. As you all know, gene editing, though a relatively recent technology, has enormous potential and is being widely applied for dealing with several challenges in healthcare and agriculture. The inventors of CRISPR-Cas have recently been awarded with Nobel Prize for Chemistry 2020. Extensive research efforts have been initiated in India also, and there's a need for enabling environments to make use of these applications of gene editing for the benefit of farmers and society. In view of all the above, this webinar is being organized to deliberate on the prospects and perspectives for this technology for agriculture, society, and sustainable development. The way we have structured this webinar is that we will be having two presentations to set the context, followed by a panel discussion by national and international experts. I am very pleased to inform you that the webinar will be chaired by Dr. R.S. Paroda, Chairman, Trust for Advancement of Agriculture Sciences, and former Secretary, Department of Agriculture Research and Education, and the Director General, ICAR. Thank you very much, sir, for being able to join this webinar and share these deliberations on a very important topic. You all must have also received an article written by sir in the Financial Express a few days back, which also reflects on the enormous potential of the technology and how we can take forward in India. So the presentations will be made by Dr. Vishwanathan Chanuswamy from Indian Agriculture Research Institute and by Dr. Donald Mckenzie from Donald Dunford Plant Science Center. And then our panel of esteemed panel, which consists of the experts, includes Professor Deepak Pental, former Vice Chancellor, University of Delhi, Dr. K.V. Prabhu, Chairperson, Protection of Plant Varieties and Farmers' Right Authority, who is also steering the formulation of regulatory framework for gene editing in India, Dr. Shubhra Chakravarti, Director, National Institute of Plant Genome Research, Dr. N.K. Singh, National Professor, B.P. Paul Chair from the National Institute of Plant Biotechnology, and Dr. Amitabh Mohanty from Corteva Agri-Sciences, Johnson, USA. So, and also I'm very pleased to inform, thank you all for, being able to spare time and being able to participate to giving presentations and your views in this webinar for the benefit of the participants. I'm also like to share with you all that very pleased to share that we got almost more than 1,000 participants have registered for this event, not only from India, but also from other countries. The profile of participants includes regulators, policymakers, research scientists from public and private sector, researchers, and other technical agencies. With this brief introduction, I would now request Dr. R.S. Paroda to kindly give his opening remarks. And before, sir, I'd just like to give a brief introduction to Dr. Paroda. He's been as I mentioned, the former Director General of ICR and Secretary there. Sir has made significant contributions in the field of agriculture in India. He modernized the Indian National Agriculture Research System, has been the President of Indian Science Congress and the National Academy of Agriculture Sciences, and recipient of many awards. It's a too long a list and I won't go into too much detail, but just to say that he received the Honorable Padma Bhushan Award also, and currently the Chairman for the Trust for Advancement of Agriculture Sciences. Over to you, sir, for your opening remarks.
Thank you, Vibhaji. Good morning. My welcome to all the distinguished participants. Indeed, it is an important webinar being organized by TIGS and BCIL, for which we would like to place our appreciation on record to the organizers. And I'm extremely pleased that this important subject, which is of great importance for not only today's but future food security globally, is being addressed today. To this August gathering to tell about importance of genetic variation perhaps is not necessary, but starting from Darwin's theory of evolution, everything had centered around variation and selection process that has gone along uh, all through the civilization. And in this process, many useful plants had been selected, and possibly that was the basis for settlement of all those who were nomads in the past, and the civilization began along the important rivers of the world, starting from Yellow to that of uh, Middle East to that of Indian, and also in case of Mexico. Uh, in this context, as a student of genetics and plant breeding, to begin with, we have been told that you need to create more useful variability than what is available for the benefit of your stakeholders, and especially the stakeholders are our farmers. And for that, you have to think that it produces more, it has resistance to biotic and abiotic stresses, and in today's context, it also has adaptation to climate change. So in that process, we need to see as to what we can do to provide yet better uh, materials and plants and varieties to our farmers. In the process, a lot has happened uh, thanks to the discovery of Mendel laws of inheritance. Then, of course, came the discovery of DNA by Watson Crick, and then also we knew that Dr. Borlaug received the Nobel Peace Prize based on the use of dwarfing genes in wheat, uh, which led to Green Revolution. And it's that time when I was doing my PhD at Indian Agriculture Research Institute, and seeds of Green Revolution were being sown. And in the past, we were all told that breeding is just hit and trial exercise. Uh, it is a number of, it is a game of numbers, and you have to try your luck by crossing the desirable plant types, whether within the available cultivated varieties or through wild and weedy materials. And in that exercise, uh, much could be accomplished, and I won't like to go into the details of those, but had those developments not been possible, probably we would have not achieved the kind of food security in most of the different parts of the world. Uh, in this context, we were also aware that even the discovery of jumping genes was given by Barbara McIntosh, but her work was recognized almost at the fag end of her life. But those were very important discoveries. And recently, it is encouraging to see that two women scientists as a team for the first time have received again Nobel Peace Prize for chemistry. One from France, Emmanuel Carpenter, and one from US, Jennifer Doudna. So I see that things have happened, things are, and this technology, I say that this is a disruptive innovation, and science will progress through innovation. And even the Green Revolution was innovation-led Green Revolution around those dwarfing genes, whether wheat or rice. And now we have options through this technology to create genetic variation at will, which was earlier possibly not easy to accomplish. So obviously, An issue before us is that this offers an opportunity, whether this can become an international public good as a technology while taking care of intellectual property rights is also another aspect which we have to consider while we discuss and try to see. It has created a lot of excitement among researchers, postgraduate students, and many scientists have come up with the useful, achievements. In case of banana, from National Agrobiotechnology Institute called NABI in India, and recently I read, maybe you have also read, that in Japan there is application now for registering tomato,
Very much.
the this technology. So the issue is how can we take benefit of it, though there are now great options. It is like, editorial corrections in any statement using English as a language. So within language, within dictionary, if you are trying to edit and making good sense of something which is already existing, then that is what is I would compare with gene editing. Uh, so we have the option, only problem comes that if we are using English dictionary and try to incorporate words from Hindi dictionary, then there may be difficulties. Just in a simple language, I would like to mention. So if it is within, we have been the breeders, we have been the geneticists, the biotechnologists, and we have been evolving new genotypes. But if they are within the cultivable genera and genus, uh, there should not be any specific concern. But no doubt, genetic engineering has offered new options for incorporating genes from non-related organisms. And in that context, if it is from bacteria or from other organisms, then as it has happened in uh, what you call as genetically engineered plants, then there had been some concern. So people seem to be now debating whether you need regulatory system uh, if you are using this particular approach, and if you are using regulatory system, then it becomes much more difficult. I feel that it should be treated simply as any other breeding method, unless you are trying to bring in genes from unrelated species. And in that context, the debate will be there. In National Academy of Agriculture Sciences, we had good discussion on it. Dr. Nagendra Singh is now with us here. He had been the leader of the working group, uh, and they have come out with various different options and guidelines. These guidelines were then submitted by us. Even I had also written to the ministries in environment and also in science, including Secretary DBT, ICR, suggesting that we need not bring it under regulatory system. In US, they have clearly come forward and cleared it, stating that they need not be under regulatory system unless there is transfer of any unrelated organism or they are transgenic in true sense. Uh, in that respect, now we have to see whether Indian government agrees to the suggested guidelines, which are currently with the GEAC, that is Genetic uh, Engineered Appraisal Committee, and I hope it will be cleared soon. And then it requires a policy decision. And above all, I consider not only policy environment, but the public awareness, which is very important. Public must be clear, because when we bring the doubts in the minds of the public, then the whole thing becomes counterproductive. And our effort here is to generate something new, useful, uh, whether it is resistance to increased concentration of biotic and abiotic stresses or climate change, heat tolerance, cold tolerance, drought tolerance, salinity tolerance, and these are all required. Even we will require the varieties which are going to do better under stress nutrient conditions, stress water conditions, micronutrient conditions, and with better quality. So ample is the scope there. I would not like to make long, my time is up. I would only like to say that I am pleased to be participating in it, and I would like to thank Vibhaji and also TIGS for the invitation to participate. And I know that there is an excellent panel of speakers and the panelists. And also, I am told that more than 1,000 participants have joined us this morning. So it is a great initiative, and let's move forward and see if we can come out with some good recommendations at the end to see as to what is the best way forward as far as Indian agriculture and benefit of this innovation to our smallholder farmers is concerned.
Thank you, sir. Thank you so much for the very wonderful remarks and setting the stage for the discussion. And this one, so as I indicated, and to start with, there are two introductory presentations that we have. The first presentation is by Dr. Chinnuswamy. Dr. Vishwanathan Chinnuswamy is a principal scientist and head plant physiology at the Indian Agriculture Research Institute. He has 24 years of research and teaching experience in plant physiology and published more than 150 peer-reviewed publications. He has been instrumental in establishing genome editing in indica rice and developed mutants of rice mutants with enhanced drought and salt tolerance. So, Dr. Chinnuswamy, it's over to you.
Good morning to all of you. Respected Chair Padma Bhushan, Dr. R.S. Paroda, Dr. Donald Mckenzie, panelists, Professor Deepak Pental, Dr. Prabhu, Dr. Subra, Dr. Amitabh, Professor N.K. Singh, and Dr. Vibha and Dr. Dasgupta, and all the attendees. Good morning to all of you. I will be making a brief presentation on introduction to gene editing and its application and the Indian initiatives so far made. So, the genetic information in the DNA, that is, all our genetic information is written in the DNA in the 4 alphabets, A, T, G, C. And these alphabets can be changed because of the mutation, and mutation is the basis for evolution, as already Professor Paroda explained. So these mutations can be a simple substitution or an insertion of nucleotide or a deletion of nucleotide. This can change the alphabets, the way in which the genetic information is coded in the DNA sequence. Then this genetic information is translated into protein, which gives us the phenotype. So the mutations occur naturally. This is because of the chromosomal aberrations that happens during the meiosis. That is, once the cell division takes place, there may be some chances of mutation because of the recombination mistake or crossing over mistakes, or during the replication of cells, that is reproduction of cells due to mistake of DNA polymerase. When it reproduces or synthesizes the DNA, there may be a mistake, or naturally there is a lot of DNA damage occurs in the natural environment, and these DNA damages are repaired by cellular machinery. So during this process, the mutations can occur. So mutations occur naturally, and these occur at a slower rate because the cell has efficient mechanism to repair it. Mutations can also be induced by using radiation or chemicals. This radiation or chemical, they cause damage to the DNA. So at any place, if there is any damage, then during the repair process, there is a possibility of mistake by the repair system, and this leads to mutation. So mutations in general, they are random and unpredictable. As explained just a few minutes back by Dr. Paroda, the mutations are the cause of evolution, and that is causing the variation in the natural germplasm. And this is the basis for breeding, that is the germplasm variation. So we initially, when the breeding started, we used the natural allelic variation that existed in the germplasm. And the later, in the early 19th century, when the mutations were discovered, and the mutations can be induced by radiation or chemical, breeders use this radiation or chemical induced mutation to generate variation, and from the variation we selected the desired mutation. And later, because it is random and it is unpredictable, selecting a desirable mutant for a desirable trait was very difficult. It was only the phenotypic approach. So later, advanced technique came in, which is called as targeting induced local lesions in genome. That is, in the genome, in any place, if an alphabet is changed, how to identify that. For that, this technology came in. By using this technology, some advancement have been made. So by using all these technologies, varieties have been released, and these are currently widely cultivated. In the phase 3 of the breeding came the transgenics and the cisgenics. And in the phase 4, in the recent phase, the genome editing technologies came in, where we can precisely make mutation in any particular location in the genome. So genome editing is a site-directed mutagenesis process to make desirable mutation in the gene at a known location. As I told, the mutations are random and unpredictable. Often the mutation may be desirable or non-desirable. So the genome editing tool offers us a technology to make desirable mutation in the precise location in the gene of our interest. So earlier, these are various technologies have evolved for genome editing. So the basis of genome editing is to create a DNA damage at the site of our interest. Once we create a DNA damage at the site of our interest, then the creation of mutant takes place due to the natural cellular DNA healing mechanism, and that during the error in that repair mechanism causes the mutation. So we need to have technologies that can precisely make mutation in the DNA. So for that, earlier the DNA binding protein fused with nucleases have been used, that include zinc finger or the tail effector nucleases. So these are protein-based techniques. Protein DNA interaction was established. So this was very difficult, as the protein has to be engineered to target the protein to a specific location on the genome. So recently the RNA DNA interaction based, the CRISPR-Cas technology came in, which revolutionized the genome editing. So here the nucleus is taken to the specific location on the genome based on the guide RNA sequence which we use. So for this, as already told, Professor Jennifer Doudna and Emmanuel Carpentier have received the Nobel Prize for Chemistry in 2020 for the discovery they made in 2013. So in brief, we have to have a guide RNA sequence, that is a 20 nucleotide RNA sequence, which is complementary to the genetic information or the gene which we are interested, where we want to create a mutation. And we have to have a nucleus, that is the Cas9 nucleus, which is an endonuclease. So we have to put this guide RNA and the Cas9 nucleus into the cell by using various mechanisms. Either we can use agrobacterium mediated genetic transformation, or we can use various other methods. We introduce this guide RNA and nucleus into the cell. Once we introduce the guide RNA and Cas9 nucleus into the cell, the guide RNA binds to the Cas9 nucleus, and the guide RNA takes the Cas9 nucleus to the specific location. Out of the millions of base pair present in the genome, the 20 base pair guide RNA takes the Cas9 nucleus to the specific location where it is exactly complementary to the guide RNA. Once it is taken to that specific place, the Cas9 nucleus makes double-stranded cut. So this is the fundamental for the gene editing. Once it is made cut, the reagents, the role of these reagents are over. So now the cellular repair machinery takes over. So in this, two type of repair takes place. One is the non-homologous end joining repair. So this DNA break is repaired by this mechanism. During this mechanism, some error occurs, so there is a possibility of mutation, indels, and or it can be addition or deletion of few nucleotides. Or alternatively, if a repair template is present, the homology-dependent repair can take place, wherein the repair template is incorporated in the mutant, the double-stranded break place. So there is a possibility of insertion or replacement of the existing template with the repair template. So based on these repair mechanisms, the mutations are created. So once in simple, like if the reagents are transferred into the cell, the Cas9 nuclease and guide RNA, they are incorporated into the genome at one place. And if our target place is here on another chromosome, or a place which is distant to the tDNA insertion place. So now the guide RNA and Cas9 creates the mutation, and the primary transgenic plants are obtained. During the process of selfing, in the next generation, the tDNA part separates and only the mutant part remain in the desired plant. So the final product is no transgene is present, and this mutation is indistinguishable from the conventional mutation that has been created by the natural way or by the induced mutation. So based on the way in which the mutations are created, these mutations are classified into three types, that is site-directed nuclease 1, site-directed nuclease 2, and site-directed nuclease 3. That is how the double-strand break is repaired. Based on that, the mutants are classified. The SDN1 type of mutation, after the nucleus cuts the DNA, the repair process is natural. So during the repair process, simple mutations are created. So these kind of mutations are known as SDN1 mutations. In the SDN2 type of mutation, we provide a repair template to repair the double-stranded break DNA. So the repair template may have one or two nucleotide change. So this leads to a precise change of nucleotide in the desired gene. So only one or two nucleotides are changed at our will in the precise location of the genome. In the SDN3, we provide a repair template. This repair template has a native part of the genome, that is homology region. Other than that, in between we have a region which is a foreign gene or which is a gene from other plant species. So this can also be incorporated. So this is useful for insertion of foreign gene at safe harbor loci. Otherwise, when you produce conventional transgenic, you have to create large number of events to produce an event that is not disrupting any natural function. So all three possibilities are available by using the genome editing. So since the SDN1, SDN2, SDN3, these definitions are precise, has been initially made by on the basis on which the double-stranded breaks are repaired. But after that, the technologies have evolved further. There is no need for double-stranded break. We can precisely change a nucleotide in the gene of our interest. So these are called as base editing. By using cytidine deaminase or adenosine deaminase, we can specifically create mutation at specific place. So there is no double-stranded break. So in this, double-stranded break repair mechanism is not involved. The another advancement in the genome editing is prime editing. Again here, we use exogenous template which is fused to the guide RNA. So it can specifically incorporate at this target loci, the mutation can be specifically incorporated by the action of the reverse transcriptase. So here also double-stranded break is not there, only a single-stranded break is made, and at this point the corrections are incorporated. So these are some of the advancement, and the technology is evolving day by day. Further, the genome editing has a large number of applications, which has been already explained by Professor Praroda. So we can create any kind of variation that leads to enhanced yield or enhanced stress tolerance or enhanced nutritional quality. So as it was told that the SD1 gene brought green revolution in rice. So this was natural mutation which was incorporated into IR8, and then it was incorporated into other varieties. So these kind of mutation can be easily created by using the genome editing technology. This is in the Kasala genotype. These mutations have been created, which leads to enhanced yield. So similarly, these mutations are applied internationally to enhance the yield. So this is one of the example where the mutation in the ABA receptor enhanced yield under field condition. This experiment was done in China. So the other examples include the genome editing of susceptibility genes, and this can increase the bacterial blight resistance in rice. So otherwise, bacterial blight is a major problem. So if we use genome editing, that is the sweet gene promoters are edited, this can lead to enhanced resistance to bacterial blight. So in addition to editing the elite varieties, we can also edit the wild genotype or the land races which are resistant to various stresses. The yield is less, so we can make precise editing to improve the yield of those varieties. These are called as de novo domestication. So this is also possible by using the genome editing technology. So these are a large number of effort that made globally in different crops for editing various agronomically important traits. So finally, in India also has significant progress in this direction. So both DBT and ICR Institute have made a lot of effort. So DBT has a special genome engineering technologies task force under which large number of projects have been given. And in these projects, now products are being developed. Similarly, in ICR also, under National Agriculture Science Fund, the projects are making progress. So few examples I will show from the Indian scientist. So this is one of the example from Dr. M.K. Reddy's group, ICGEP. He has edited 2 genes in the MTU 1010 background. So this led to increase in the 25% yield. That is when the DEP1 gene is mutated, or in case of TB1 gene, when it is mutated, the yield increase was about more than 70%. So these are in the greenhouse studies. So we can evaluate in the field condition, they have a lot of potential. In IRA also, we have developed mutations that enhances the salt tolerance of the MTU 1010 by simply editing one of the genes, which is known as drought and salt tolerance. This NABI example by Siddharth Diwari was already given by Dr. Paroda. So editing of lycopene epsilon cyclase resulted in enhanced beta-carotene content in the pulp of the banana fruit. So this can increase the vitamin A level in the banana. And similarly in TNAU also, they have developed aromatic mutation in the ASD 16 background. So there is a lot of potential for genome editing application in India, and India has made quite a good progress. So the conventional mutagenesis methods are random, unpredictable, and it causes multiple mutation. So then transfer of these mutation to desirable elite genotype, it is time consuming and associated with linkage drag. Whereas genome editing is a precision mutagenesis tool, it requires less time, no linkage drag, and it has great potential for revolutionizing the crop improvement. So I thank the TICS and BCIEL, Dr. Vibha Agusha and Dr. S.K. Dasgupta for providing me this opportunity. I thank the funding agencies for the work supported to our lab. Thank you very much.
We will now move on to the second presentation, and I invite Dr. David Mckenzie. Dr. Mckenzie is the Executive Director of the Institute for International Crop Improvement in Donald Danforth, the Plant Science Center based in US. Dr. Mckenzie is an international expert in regulatory systems for agriculture, including environmental risk assessment, biosafety, food safety assessment, etc. And he's managing at the present, he's managing all the partnerships and programs which are dedicated to translating the key discoveries in plant disease and pest management, genomics, advanced breeding, and nutrition, etc. So that these technologies can reach to the farmers. Navigating through the practical safety and the regulatory processes is his area of expertise. So we look forward to hearing from Dr. Mckenzie on the enabling environment, the need for the enabling environment that is required for taking forward these technologies. Over to you, Don.
Okay. Good morning and good afternoon and good evening to everyone wherever you happen to be today. I want to thank the Tata Institute for Genetics and Society and Biotech Consortium India Limited for this opportunity to participate in this webinar on genome editing for agriculture and society and sustainable development. My thanks also to Dr. Paroda for his opening remarks, and to Dr. Chinnasamy for very precisely and thoroughly elaborating on the technology and on the applications of genome editing to improving crops in agriculture. So, as has already been mentioned, for thousands of years, humankind has initially through selection and later through crossbreeding sought to modify the genetic makeup of plants to improve their characteristics for use as food and fiber, and also as feed for livestock animals. A major goal of plant breeding is to continuously make use of and then to increase genetic diversity. And has already been mentioned by Dr. Chinnasamy, the first attempts to increase genetic diversity by technical means were in the 1930s, using either radiation or chemical mutagens to introduce randomly mutations into the plant genome. And that was followed by intense selection to identify those few progenies that would have the desirable traits. With new technologies around genomics, we were able to associate genetic sequences and genes with plant characteristics, and then could move into using marker-aided selection and other so-called smart breeding techniques. Later on in the early to middle of the 1980s, and then with their commercialization in the middle of the 1990s, was the introduction of transgenic plants and crops, so-called genetically engineered crops. And these products have not only improved farmer livelihoods everywhere where they've been adopted, but they've also decreased the environmental footprint or the environmental impact of agriculture. So now with the introduction of genome editing, breeders can now more precisely target exactly where in the plant genome we want to introduce mutations or to introduce specific changes. So very practically then, genome editing can be considered to be an extension of mutation breeding. And the vast majority of plant breeding innovations, or PBIs as I'll call them, are those of the SDN1 and SDN2 type that were created and contain mutations that are really indistinguishable from the types of mutations that could be created using classical mutation breeding techniques. It's just worth pointing out and acknowledging a recent opinion by the European Food Safety Authority, EFSA, and released their opinion just past October, stating that in their view, products of genome editing, specifically those that are SDN1 and SDN2 types, are no more hazardous than those products produced through conventional breeding methods. This is something that we've always, I think, known since the late 1980s in our evaluations of genetic engineering and its products, in that it's really not the technology that's used to produce it, but it's the characteristics of the final product that determine risk. So that recent opinion by EFSA is a good reminder that all plant breeding methods, regardless of the technique, have some potential for giving rise to unintended effects. And you'll see that many of them, in fact, nearly all of them, we have found a way to regulate them using traditional agri-food regulations, what we would call regulated as conventional. It's really only those exceptions, products produced through recombinant DNA technology that have in the past 25 years or so resulted in the elaboration of special regulations, special biosafety requirements and regulations to deal with those products. When in fact, if we look at the plant breeding technique that is the most likely to give rise to unintended effects, that is classical mutation breeding, we find that we can deal with those products perfectly fine using conventional agri-food regulations. So just also a reminder to ourselves that the conventional agri-food regulatory system involves a whole array of different regulations geared at managing seed production, crop improvement, crop production, the use and distribution of grain, the use of plant products in food manufacturing, and also of distribution and marketing. So it typically involves multiple agencies operating at different levels in government, at a national or central level, at state or local levels. It also involves international actors, such as those involved in the International Plant Protection Convention, the International Seed Trade Association Codex, and so on, that set standards internationally that have to be met locally and regionally. We have also regional trade organizations that similarly set standards. So the blend of developers that are working on plant breeding innovations is strikingly different than the mix of developers that have successfully commercialized or introduced genetically engineered plants into the marketplace. As we can see from these data that were collected in the US and based on an analysis of the MI regulated applications and the determinations coming out of them, we can see that more than 90% of the developers are really coming from the public sector, from universities, from nonprofits and government centers, and also from small and medium-sized enterprises. Only a small proportion are coming from the transnational corporations or the MNCs. If we were to look at the current collection of commercialized genetically engineered crops, the situation would be entirely different. It would be exactly the opposite, where only a small proportion of developers are coming from small organizations or the public sector. In fact, there are really very few examples that we can think of public sector products that are actually being grown or actually are in the hands of farmers. 2 or 3 examples would include virus-resistant papaya that was developed by USDA Agricultural Research Service and released in 1998. Another one would be BT brinjal, which was approved in Bangladesh and was released to a very small number of farmers beginning in 2014 and is now being grown by probably much more than 30,000 farmers or so. And my final example is a product that is just about to be released to farmers, and that's BT cowpea. So that's transgenic cowpea that's resistant to Maruca pod borer that was approved for commercial release in Nigeria in January of 2019. And the first registered variety is now being grown by farmers in demonstration plots with broader commercial release planned for this coming year. That's a really small number of examples. And if you think about the tens of millions of dollars that governments and donor organizations have invested in these kinds of projects, you might say it's a pretty discouraging return on investment. And the existing regulations for genetically engineered crops have largely focused on potential risks and have not focused on trying to balance risks and benefits. They've added increased complexity. They've added vastly increased costs, and all of these have significantly disadvantaged both public sector and small developers. So these weak policies and burdensome regulations have meant that only a few traits have made it to market with respect to genetically engineered crops. A small number of traits that would include mainly herbicide tolerance, insect resistance, perhaps to a lesser extent, modified oils or drought tolerance, and only then in a handful of crops, primarily maize or soybean or cotton and canola. That represents the majority of what's in the marketplace now. And these products have mainly been introduced by, again, the large organizations, mainly transnational corporations, who have the resources and ability to navigate multiple complex regulatory environments, something which is really a challenge and nearly impossible for small developers. So to really make and realize the benefits of PBIs and to make the product more accessible to public sector developers, in essence, to democratize the technology, we need to simplify the regulatory approach so that we can end up with a broader diversity of traits and a broader range of crops for the benefit of smallholders. So I want to spend just a few minutes talking about the evolving regulatory landscape around genome editing in plants around the world. And I think at the outset, it's worth noting there's this country colored in blue, which is different than all the others, and that's Canada. And it's only to make the point that its regulatory framework has not had to evolve at all with the advent of genome editing. And that's because since 1993, Canada's regulations have been based solely on the novelty of the new plants or new foods or feeds that have been developed and not on the process used to create them. It's also worth noting that we have a collection of countries in South America that have shown very strong leadership over the past few years. And this has really stemmed, I think, from a ministerial declaration that came out in September of 2018 from the Agricultural Council of the South, agricultural ministers, in essence, that represent a collection of countries in South America. And in that declaration, they stated that it was appropriate to avoid making arbitrary and unjustifiable distinctions between farm products delivered by gene editing and products delivered through other breeding methods. And they would work jointly with other countries to avoid non-science-based trade barriers to farm products. So those countries in South America have, in essence, taken the decision that gene-edited products that have targeted deletions or targeted edits, so SDN1 and SDN2, would not be regulated like GMOs. However, gene-edited products that would be classified as SDN3 that have inserted foreign DNA would be regulated like GMOs. We have a small number of countries, the US, Australia, and Japan, that are a slightly different shade of green. And this is because they're partial positive opinions. So it was mentioned earlier about the US Department of Agriculture's new secure rule and the fact that USDA has made some categorical exemptions for gene-edited plants that contain a single change. So that would be a change resulting from cellular repair to a targeted DNA break, so SDN1. And that's in the absence of any template DNA. A targeted single base pair substitution, or the introduction of a gene known to occur in the plant's gene pool, or a change in a targeted sequence to correspond to a known allele, so allele replacement. So plants that contain or are those kinds of modifications are exempt from regulation. And here I think it's worth pointing out that there is a difference between exemption and deregulation, and sometimes they get confused. And so in this case, we're not talking about deregulation, i.e. taking something that was regulated and now deregulating it. It's about creating a category of exemption so the products are not regulated at all at any stage. And this process is really involves self-determination by the developer. The developer decides whether their product meets the qualifications for exemption or not. The developer may voluntarily go through a consultation process to confirm that their product meets an exemption, but that's not mandatory, that's purely voluntary. In Australia, it's a partial positive opinion really because Food Standards Australia New Zealand is still working out what their process is going to be, and OGTR has decided that they would not regulate SDN1 like GMOs, but that SDN2 products would still be captured under the Gene Technology Act. I should point out that in the US EPA is in the process of finalizing its rule on how it's going to deal with gene-edited plant-incorporated protectants, but has not yet done so. And of course, the Food and Drug Administration has an entirely different process for dealing with bioengineered foods that's not expected to change with gene editing. There's a large block of countries in Europe, colored in red, that have essentially taken the decision since the summer of 2018, to regulate all gene-edited products just like GMOs. And this has, in the past, Europe has had a sort of an outsized influence, if you will, on other countries with respect to how it's dealt with regulations around genetic engineering. And that's been particularly evident in countries in Africa, many of whom are concerned with how European regulations might affect their exports to the European Union. It remains to be seen how Europe's stance on gene editing will affect developments in Africa. But it is encouraging to note that the discussions ongoing in both Kenya and in Nigeria have resulted in guidelines for gene editing that are almost finalized. And they are, in spirit at least, if not in total actuality, following the intent of what's been happening in South America. That is, they would find a way to not regulate products that would be classified as SDN1 and SDN2, would only regulate those gene-edited products that contain inserted foreign DNA, so SDN3. It's likely that the guidelines in both of those countries will be finalized in the upcoming year. So we have other countries that are colored in yellow where discussions are ongoing. India, of course, being one of them, China another, and the Philippines is one that I'll spend just a little bit of time talking about. So at present, The Philippines regulates genetically engineered plants and GMOs under a joint departmental circular that involves the Department of Science and Technology, the Department of Agriculture, the Department of Environment and Natural Resources, the Department of Health, and the Department of Interior and Local Government. So 5 different departments and agencies. This system has been in place since April of 2016 and is the result of a Supreme Court case and a Supreme Court decision that was rendered in December of 2015 that essentially struck down the existing regulatory framework under the Department of Agriculture's Administrative Order number 8. So under the Joint Departmental Circular, it has adopted the definition of GMOs that a GMO contains a novel combination of genetic material obtained through the use of biotechnology. But it does not really elaborate more than that on what is meant by a novel combination. So beginning in the spring of 2019, the National Committee on Biosafety for the Philippines started a consultative process by creating a technical working group with a remit to prepare a policy for the Philippines to adopt on how it should be dealing with the products of genome editing. They had workshops, they had consultations, and in April of 2020, they came out with a draft resolution defining how they would deal with gene edited products. They defined and used some new terminology such as plant breeding innovations and they clarified what was meant by a novel combination of genetic material. So that's a combination that could not be achieved through any conventional breeding process. So if it's not possible through conventional breeding, it's a GMO. If it's a non-GMO, then it should be regulated as conventional. They developed a pretty elaborate decision tree on how to deal with various products of modern biotechnology. 2 of them would be fall under the classic GM regulatory framework, and that would be transgenic products and also SDN3 products that have a foreign DNA insert. Products that would be of the SDN1 and SDN2 type, as well as products that could contain cisgenic inserts, would not be regulated as GMOs and would be essentially regulated as conventional breeding products. So in their plan for implementation, or how they would envision implementation, then contained work, that's the plant transformation, selection, evaluation in a growth chamber or a greenhouse or a screenhouse, that would be regulated as it is now under the Department of Science and Technology. However, once the exogenous DNA is bred out, the Department of Science and Technology would grant a certificate confirming no novel combination of genetic material and hand it over to the Department of Agriculture, who would then essentially regulate the product as it does for conventional breeding products. So the latest status on what's happening in the Philippines is that there are 7 cabinet secretaries who have to sign off on the draft resolution, as well as 6 other scientific members of the Biosafety Committee. I understand that perhaps all but a couple of the cabinet secretaries have signed off, and there's every expectation that the remaining signatures will be received in the near future. So we can look forward, I think, to sometime in 2021 of having the Philippines finalize its approach on genome editing. So just some final thoughts. You know, the challenges are great and time is short. I think we're all aware of the impacts of climate change on facing plant breeders in the next number of years. And if we think that, you know, of traditional plant breeding cycles, development cycles being on the order of 8 to 12 years in duration, even much more if we're talking about genetically engineered crops, we really don't have a lot of time at all. And you know, the impacts of climate change by 2050 in South Asia are estimated to result in a 15% or more decline in productivity and a more than 30% increase in prices. So there's every incentive to use every technology we have at our disposal in a very agnostic way to solve important problems as quickly as we can. And we have a lot of experience in dealing with plant biotechnology products by now. And I think one of the lessons is that we should follow the science, capture the learnings of more than 3 decades of regulating GMOs, and think about how we can take perhaps a different path that will benefit not only small and public sector developers, but obviously smallholder farmers as well. And to realize that, you know, nearly all of the PBIs that are being produced now through genome editing can be quite reasonably regulated as conventional and not under GMO regulation. So with that, I thank you very much.
Thank you. Thank you, Don, for a very nice presentation and giving us the overview of the regulations, regulatory processes around the world, and your thoughts on how it should move forward. We will now move on to the panel discussion. And as I mentioned in by starting the meeting, that we have a very esteemed panel of speakers. And the way we've structured that, we would request all the panelists to kindly share their views initially for 5 to 7 minutes on the theme of our webinar today. And then we will have a kind of a specific question-answer based discussion. So just to move forward, I will just repeat and introduce the panel again. We have Professor Deepak Paintal, CSIR Distinguished Scientist and former Vice Chancellor of University of Delhi, Dr. K.V. Prabhu, Chairperson of the Protection of Plant Varieties and Farmers' Rights Authority, Dr. Shubhra Chakravarti, Director, National Institute of Plant Genome Research, Dr. Amitabh Mohanty from Corteva Agrosciences, and Dr. N.K. Singh from the National Institute for Plant Biotechnology. So I will now take a round and one by one request all our panelists to share their views. And now may I request Professor Deepak Paintal to kindly share his views.
Good morning to all the fellow panelists. We had 2 very good presentations by Vishwanathan and Dr. Don Mckenzie, which I think have taken us through both what the technology is and what are the regulatory issues. I would like to make a few points quickly. You know, we are going through difficult times, and I think people are getting better understanding of the threat of pathogens due to this COVID pandemic. And it is not generally appreciated that plants also have a large number of enemies in terms of pests. Pests in FAO's definition includes both insect pests, pathogens, nematodes, bacterial pathogens, and so on and so forth, and weeds. And we have been lackadaisical on appreciating this, how strong this threat is, because when you get a plant wheat variety with resistance to rust, or you get rice variety with resistance to bacterial blight or some of the other diseases, it is not a national news. It's not in the common domain that how much excellent work has been done on protecting plants through genetic interventions. These could be through sexual hybridization if it is from the primary gene pool. It could be from the secondary gene pool. And we know that it is a very difficult process of taking genes from the wild species and introgressing them into crop plants because of the linkage track problem and so many other problems. But valiant scientists have worked very hard to bring these genes, but the appreciation is very little in the society on protecting the plants. But now I think we should utilize this new development of everybody coming to know through the pandemic how much threat pathogens are to human species, but not only to human species, but to like Gemini viruses are creating havoc and shaving off a lot of yields in a number of our crop plants. So we need all kind of technologies to combat these pathogens of plants. Wherever genetic intervention can be done, it should be preferred over agrochemicals. But there are areas like weed control which can be only dealt with agrochemicals. So we need all kind of technologies. But public sector like CG institutions or national agricultural institutions getting in a big way into the area of resistance to pests and pathogens. And the farmers take up these technologies very fast, as we can see from PT experience or from herbicide tolerance, where things were not even released properly and the farmers had started taking up. So the farmers like these technologies, but they don't know that how much of these exist and where they exist. The other thing I would like to point out is the current agitation by farmers which are deeply stuck in the wheat-rice cycle. And we all know that this is not sustainable. It is taking away huge amounts of groundwater, and we need crop diversification. Now just imagine if farmers had another crop in lieu of rice, which would give them as much income, but use much less natural resources. I think farmers will take it up without any difficulty. I have this major point to make that we have not done enough on the seed side. We have not strengthened our crops for resistance against pests and pathogens, because alternate crops, people talk about maize, people talk about soybean. They are also susceptible to diseases that the farmers will not get more than 1 ton per hectare. So the farmers which are used to 4 or 5 tons of rice on an assured price are very reluctant to move to alternate crops. So we need to strengthen our alternate crops like oilseed crops, legume crops, and make them resistant to diseases and pests. And that is where the big challenge is in terms of diversification. Now, if you look at it, the last point I would like to make is, look at the investments which India has done into irrigation. The large dams, which have been all public funded, canal system, and then you take the tube wells, which are the mainstay in many parts of the country in terms of irrigation. There's so much private investment. So huge investment. The European countries would not have made that kind of investment because they are naturally blessed with good rainfall, except in the southern parts of Europe where there is some irrigation being done. So these countries have not spent anything, but India, China, and many other parts of the world, like in California and the United States, have spent a lot of money on irrigation. India has spent certainly a lot of money on irrigation. We are using fertilizers, and I don't believe that our fertilizer usage is maybe not optimal, but it is not excessive if you compare it with what is being used globally. So we are heavily subsidizing fertilizers. So we are spending a lot of money on irrigation and on fertilizers, but we are spending very little in terms of R&D into seed. And that is a tragic part in our whole approach to agriculture. We need all the strength in the seed, and we need all the technologies that are available to strengthen the hands of our farmers so that they can go for low input, high output agriculture. So I am all for the new technologies. The STN1, STN2 should not be regulated at all. In fact, our basic sciences are suffering because we have not been able to create enough mutations for structure-function relationship. And STN1 should be a normal technique in all the laboratories to do structure-function relations and to find out what the genes are doing in a plant. And finally, I would say that in resistance breeding, we need genetic engineering to bring in genes from the wild, and we need genetic genome editing for particularly removing the susceptibility causing genes and removing the antefeedants and so on. So these technologies are mercifully have come our way to strengthen our cropping system, to strengthen our crops, and not using them would be, in my view, would be biggest blunder that anybody can commit. So I've said enough. If there are many other points that will come through, we can discuss them later. Thank you.
Thank you, sir. Thank you very much. And we will move on. And we have Dr. Shubhra Chakravarti. I would request Dr. Chakravarti, Director NIPGR, to kindly make her comments.
First of all, thank you, Dr. Bibha, for inviting me into this to just give a little bit of my thoughts about gene editing and its use. So I think I would like to flag two things. One thing is that what is the need today when we are moving towards the maybe 2030 vision that the world has to achieve, and India is anyway part of the world. Scientifically what we require for the plant improvement, and then maybe the second part is how can we use this gene editing to basically cope up with this kind of a challenge. So coming to what is needed, if we actually see either from the perspective of our nation, that is Indian side, or if we want to see the vision document that has come through for the Sustainable Development Goal, two very important criteria as far as plant genomics is concerned, and as far as the plant can give away for the human benefit, is two things. One is that the climate change, as we all know, is rapidly changing. At the same time, we all know that someone mentioned just before what we require by 2050, that we need to produce more as well. Although today India is a self-sufficient nation, however, keeping in view the next 10 years or 20 years, we need to still produce more. And I think there is an ICR Vision Document 2030, which also talks about possibly doubling even our cereal, our legumes, our vegetables and everything. So if we have to come back with that, when we talk about the climate is changing and what are the threats, in that case, largely it comes from a disease. And in that case, if we look at why this disease is important, there is a commonality between human disease and also the disease that plants are going through. All these microbes, because of the co-evolution, they are changing rapidly, and there is one thing that is antimicrobial resistance. We always talk about antimicrobial resistance in case of the animal or human, but if we really think deeply, what we find in case of the plant as well, basically in our all field, what are the fungicides, what are the pesticides that we are using, all came from one particular molecule, and that was somewhere in 1930s or 1940s, and that is azole derivatives. So because we have only azole derivatives, and till so far we are using that, the AMR for even plant pathogenic microbes are tremendously difficult to control. Therefore, this disease, not only for human but also for plant, is a very, very important area that we need to look up for coming years. Although I do not say that abiotic stresses are not important. Definitely they're important because the climates are changing. Global temperature is very important. Drought is very important. Salinity very important. But on top of this, I think I'm very concerned about this AMR. Now, coming back to the second point, which is also flagged in the SDG, and if we look at, we need to produce not only the quantity, but also nutrition. So nutritious food is very, very important. And if I little bit flag a data that comes from the study of the WHO, as well as from the study from other international organizations, where it has been shown, if you actually see what has happened for last 50 years and now where we are moving. Initially, if we think about human diseases, it used to be infectious diseases, where earlier used to be rampant, which used to be very important for human health. But in last 5 years, the whole shift is towards the NCD, that is the non-communicable diseases. And if we try to see, and also the maternal and neonatal and nutritional disorders. So if we try to see a little bit of a data, which I pull out from a document from 2017 document, at the global level, we still have a 60% of the disease burden, which comes today from NCDs. And 28% is from communicable or maternal or neonatal or nutritional disorders. So therefore, these are two very important issues that I want to flag. My first point, that we must give importance that how do we come up with and decide about MR in the plant so that we can make more productivity, so that cope up with demand of more food at one hand, and on the other, how do we actually make more nutritious food? And you all know that if you see and look at the 2030 vision document of the SDG, 3 of the 17 sustainable goal directly is related to zero hunger, good health, well-being, and life on land. And therefore, plant plays a very, very important role into it. And we cannot neglect the plant improvement, and in that case, if we see about the nutrition scenario, we have kept on talking, but still, if I see the data which comes from 2017-2018 documents coming from global burden disease data, or if you see any of the data which is coming from WHO, in every case it says that we need to tackle this kind of over and undernutrition issue, and which are all related to plant-based food, because 70% of our food over the world comes from the plant, and therefore we need to look for the quality angle, that is a nutrition quality angle. So these are the two major issues I think we need to address as we move along. And if I now switch to the technology, I think Professor Paroda told, and also the earlier speakers have pointed it out, we moved from almost 1930 on till last 50 years, possibly, have seen the mutation breedings and also the transgenics with the Epoch science paper, which came in 1983 of the gene technology, where we could make the transgenics. However, today we have a very, very nice technology, and that is gene editing, and therefore which takes care about all the concerns that had been raised by the transgenic approach. And if we really use, I need not to elaborate because we had such a nice presentation by Dr. Viswanathan and also by Dr. Donald. So SDN1 and SDN2 definitely, in my opinion, need not to be regulated. What I know that already there has been groups and people have worked, and Professor Paroda mentioned that the document of the Indian document for regulation is already with the DBT. If I now try to see how can we utilize this new technology of gene editing for the betterment of our crops for either quality purpose or quantitative increasing, then I would think, over the years, also the technology has changed the science of plant, or even in case of animal, whatever we say, from reductionist approach of looking at one gene by one protein, which happened in last 50 years, to find out useful genes, useful proteins for betterment of genotypes of ours. But today, we stand in an era where it's all genomics, all different omics, proteomics, metabolomics, ionomics, and many other omics. And if you look at the data that has generated and has been deposited, terabytes and terabytes of data in the public domain. The best part is all data is in public domain. And therefore, how do we now utilize this knowledge that has been generated over I would say last 10 years effort after the genome sequencing have come in. And in that case, with these data, how do we utilize this data? In that case, gene editing would be a very, very important tool. National genotyping facility, and also in India there are mission programs, there are programs on like Earth biogenome sequencing. We are going to generate again lot of data in next 5 years. So then how do we use them? So in that case, gene editing would be a very, very important technology by which we can actually, whatever knowledge we would gather for let's say making productivity more, making pathogen resistant crop, making nutritionally improved crop, we would be able to actually know not only the basic science, but also to apply it on a translational mode in the field. It is a very important technology that we must keep in mind. And I think in the coming years, with this gene editing, what I'm seeing, and also Dr. Donald talked about it, global situation, I hope that Indian policymakers and also, Indian government would be giving a very safe document wherein this SDN1 and SDN2, in my personal opinion, may not come under regulation. Although if we can actually think carefully for SDN3, which possibly, in my opinion, something which replaced the other way transgenesis. In that case, in SDN3 also, we are not going to put any kind of antibiotic, which was a major concern for the transgenic approach. And also in that case, it is a precision insertion of one particular gene. So maybe in future, we need to think on a more open angle that can we also take SDN3 under a kind of no regulation situation. Maybe little more data need to be generated before we do that, but definitely SDN1 and SDN2 is quite safe, as we heard from earlier speakers, from many global documents, and we need to adopt this for understanding biology, understanding plant biology, structure function relation, and at the same time to translate this in the field for which whole nation is looking forward, so that not only India, globally, we could actually join hand and make the world a safe place. And in that case, I think these are few points that comes in my mind, and I think gene editing is a very, very interesting tool that has come to our use, and we must use it cautiously but judiciously, and also take it in a positive way. Thank you.
Thank you, Dr. Shubhra, for very, very positive remarks, and also the need for the technology. We will now move on. Actually, Dr. KV Prabhu is unable to join because he's stuck with a meeting in Krishi Bhavan, but he has very kindly in his remarks in a video.
Good morning, everybody. First, my apologies for not being able to participate in this webinar virtually, and I am submitting this pre-recorded video version. Respected Professor Paroda, my very dear senior colleagues Dr. Pentel and many others who are participating in this webinar, I am surprised, not surprised actually, to see that more than 1,000 people have already registered, as Vibha told me yesterday, for this meeting, which is really overwhelming response. At the same time, I do feel that I am missing this direct contact with each of you to listen to you and interact with you and answer any queries while I make my panel points related to the issue in question. Plant breeding today has been benefiting to a large extent through modern genetic technologies, and this breakthrough, which Emmanuel Carpentier and Jennifer Dodha gave a few years ago, has been a wonderful boon in more respects than one, because as a plant breeder, which I have been practicing for more than 3 decades, I used to look for the character which I am aiming to improve and look for an option available in the genetic resources belonging to the species wherever, whether in India, abroad, or germplasm, or any other material which resembles this cross species which is crossable to it, a wild relative, whatever. So look for an existing trait which is going to be the part of my objective, which means we are going by already created mutation, either naturally or by any other agent which I might have used, such as irradiation or chemical mutagenesis or cytological deletion, or any of those possible changes that we can do by interacting with the genome, by pressurizing some changes to happen, some deletions to happen, so duplications to happen, or even interactive genetic fragments which will prevent an expression of another gene which I don't want to express. So these are the changes which I am looking for in existing plants. The genome editing has enabled me now to not only think of the character which I want changed, I also can imagine the extent to which I want to change, and to see that the changed plant is made available to me at the time when I require it for enabling the change in the variety which I am breeding for. All this would not have happened minus the genome editing technology. So it is a wonderful boon. Both Emmanuel Dodha and Jennifer and Emmanuel have done this wonderful exercise for the benefit of the plant breeders at large. What is required now for plant breeders to use this with interaction that is required to be established between the molecular biologists and the plant scientists who are properly trained to make this happen without leaving any element that may cause risk, as is apprehended generally. As it happens with any other new technology, people at large generally do not accept any change without being informed, without being clearly communicated that the change is not going to harm the society at large, the environment at large, animals and human beings, or the health of each of these in general. So the same thing has to happen by making the molecular biologists or the other plant biotechnologists and the breeders coming together and working on the same plant with the character in question that needs to be modified. I'm sure Chinnuswamy and David Mckenzie are going to explain to you, or will have explained already with their perspective on what the technology can do. And it is something that has come in. And I regret that I am not now in an active working phase to make use of this to the best element. I've used all the other sources of mutations that could be happening, wild species, chemical mutagens, physical mutagens during my career, and even alien species based crosses to delete or remove the chromosome by aneuploidy, etc. I've done all of this, and made use of it. But now that this opportunity is made available, my younger friends who are breeding should make use of this. Let's believe the government, which is sitting on related parameters of this particular technology, like any other technology, will do the needful and give back to the society the best gained advantage that plant breeding could have ever done from my perspective, because I don't see that there's any more change in the technology or advancement in the technology that can be better than this, other than the regulatory phenomena or the other results which are coming out of the tool that carry any chance of causing a risk, which I explained just before. So that's why this benefit which is available now to us should not be lost and made best use of. Thank you so much and have a great day today in looking at this in the webinar. Thank you.
Thank you. Thank you, TIDS team, for displaying this. Dr. Prabhu is leading the effort in developing the regulations, so his remarks have been very important. Another very important stakeholder, Dr. Paroda mentioned, and also Dr. Pentel mentioned, that National Academy of Agriculture Sciences has been very active in putting forth their perspective on the regulation of this technology and other issues. So we have Dr. N.K. Singh with us, and he's been instrumental in putting this brief together. NAS has come out with a brief on regulatory framework for genome edited plants, and hence we requested Dr. N.K. Singh to kindly share the NAS perspective to the gene editing. Over to you, sir, for a brief presentation. Yes.
Good morning, everyone. Respected Chairman, Dr. Paroda, Dr. Dan Mckenzie, Professor Pentel, and other esteemed panelists. Plus, I can see more than 550 people have joined this particular discussion. So welcome to all of you. I will be very brief because we are running short of time. And most of this thing has been said about this technology. I have just 4 points. First is the NAS policy interventions and gene technology. Then genome editing, the most precise breeding tool, as had been discussed, so I will cut short that part. And also the applications and promise of this technology. Then I will especially spend a few minutes on the NAS policy brief and regulatory framework and the recommendations that have come out of that. Well, at the outset, let me first acknowledge this has come out of a very intensive discussion. There was a roundtable discussion earlier this year in the NAS, which was chaired by the Dr. Mahapatra, Secretary DGICR, and co-chaired by Dr. Paroda and Professor R.B. Singh, past presidents of NAS. And the committee was there, which met several times afterwards to develop this policy brief. Dr. Professor B.D. Singh, Dr. Prabhu, Dr. A.K. Singh, Professor Bansal, Bhagirath Chaudhary, and Vibha Ahuja, among all. And we had a large number of esteemed discussants in that, including Professor Pentel, and there were progressive farmers and the industry representatives. So I'd like to acknowledge this has come out of a very broad discussion. And then it seems that what Dr. Don Mckenzie has presented, the global scenario, and what has been expressed by the different panelists, a kind of consensus is emerging, actually a global consensus is emerging, which is based on really common sense and the nature of the technology. And I will come to that. The NAS recommendations actually are in tune with that. So the NAS, the National Academy of Agriculture Science, as you know, is the think tank of the agriculture scientists, and they advise the government at various levels on the policy issues. And they have been doing that for the GMO also, which is the gene technology, which is guided by the regulation under the rule in 1989 of the Environmental Protection Act of 1986. So India has been there right from the very beginning. Department of Biotechnology was established in 1985. And right from the beginning, the government and the research management has been really proactive in these things. And we have a very stringent regulatory mechanism for that. But at the same time, there has been opposition to this technology by various quarters, not only globally, in India also. And this is situation in there are many groups, conservative groups, maybe there are vested interest groups are there which are very strong on this thing, opposing the genetic modification of crop plants, and genetic editing, genome editing also is on their agenda. Now the NAS has been doing the policy advocacy right from the beginning. In 2003, the biosafety of transgenic rice, the policy paper was brought out. Then in 2004, transgenic crops and biosafety issues related to their commercialization in India. And then later on, more recently in 2011, we had a detailed discussion once again when this BT Binjal was put on hold. Again, these scientists were very much concerned, and the detailed policy paper came as biosafety assurance of the GM crops of India. Then so much so that the academy has pleaded in the Honorable Supreme Court, because at that time there was an impending ban by the court for the research on the GMO. And that moment the academy pleaded in the Supreme Court, and it is all under discussion and debate only. And that much seriously the academy has taken this issue. And further, the two policy briefs have been published. One by the discussion was chaired by Professor Sathan, and the policy brief came by Dr. Mai, Dr. Prabhu, Sharma, and then this year we had an intensive discussion on the genome editing, and this very good review is presented in this particular document on all aspects of genome editing. And after thorough discussion, I will not go into details. The technology has evolved. The basic crux of the technology is that it is free from any foreign DNA, and also it is more targeted to a particular region of the genome. And that has become possible because of the availability of the genomes of many of these crop plants. Because the knowledge of genome is important to design the guide RNA which is free from off-target is very important. The technology has evolved over the years, starting from 1987 when the CRISPR was discovered, and then its role was, after nearly 20 years, its precise role was deciphered in 2007, and it was, it started using as a genetic engineering tool just 5-6 years back. So I'm not going into detail. Has been told by Dr. Biswanathan very nicely. The technology is so important that the number of patents has grown very exponentially during the last 10 years. Applications you have seen already. We can create herbicide tolerant crops. We can create waxy maize and endosperm with anthocyanin in the rice, and also virus resistant non-transgenic cucumber can be created. Knocking down of sweet genes have given immunity to bacterial leaf blight. So it is such a attractive, such a promising, revolutionary technology that very quickly, I think we should get our regulatory aspects right. Otherwise, you will miss out on this technology. And the number of products have been freed from exempted from regulation in USDA. And Dr. Mckenzie has given you this thing, an account of the global scenario. So it is coming up in a way that SDN1 and SDN2 should be free from this regulation, and SDN3 is the one which should be regulated in some way. Now there are few recommendations came from the NAS policy brief. There are 15 points. I will go in 3-4 minutes here. The first one was that since the India is a signatory to the Cartagena Protocol, and so it is governed by the EPA Act of 1986. And since the genetically edited organism, they start with the genetic transformation. So this will be also governed by this same rule. It cannot be away from this rule because we are starting with genetic engineering. And according to definition, this falls into this category. But the product is free from foreign DNA. So in that light, the whole discussion went on. And the second point was that NAS group felt that this was based on a draft guideline which was circulated by Department of Biotechnology, which has clubbed all the animals, plants, and human cells together in one document. And it was rather elaborate and probably asking for too much data. So we felt that the first thing was recommended is that we should have the disaggregated differentiated approach. There should be separate guidelines for animals, plants, and the human biomedical things. Because their level of complexity and safety risk are different. Then the fourth point was that we said that we should have a standard categorization like SDN1, SDN2, and SDN3, which has been described already very nicely. And the committee recommended that the final products of SDN1 and SDN2 developed through genome editing are free from any foreign DNA, and they are indistinguishable from the products developed through conventional breeding, which is done using either natural genetic variation or induced mutations. So therefore, it is not necessary, and also it is not possible. Because if there is no foreign DNA, how will you know that it was developed through genetic engineering, genome editing, or through conventional breeding? Even if you want to regulate, you cannot regulate because there is no footprints of genome editing there. So then it was very strongly agreed, and as around the world, the same thing is happening, that SDN1 and SDN2 need not to be regulated. Now the sixth point was that it is a precise and targeted mutagenesis tool. It's just like another plant breeding tool, but it is very advanced tool. It saves time and increases the speed of developing traits in the plants. And they do not carry, this point has been emphasized in more than one way, that SDN1 and SDN2 need not be regulated for the regulation of the risk assessment. And then how we go about the regulation, because the rule 1989 says that any product of genetic engineering must be regulated. So then people came out with this exemption under that rule, the rule 20. There was an exemption which has been granted as an exemption to the pharma products, already has been granted, and we have suggested that this similar exemption can be given to genome edited plants of SDN1 and SDN2 under the existing rules. So there is no need to make a new act and a new thing, which will take a very long time, and it may not see the light of the day. So the other point was that important point, because it starts with the recombinant DNA technique, you have to do transformation, do selection and crossing, and make plant free from the transgene. So one must do registration with the IBSC with information to RCGM. The group has suggested that only this much initial registration with IBSC, that in Institutional Biosafety Committee, and information to RCGM is adequate for SDN1 and SDN2. And there is a flow chart for that thing. Afterwards, this can be taken, the product of SDN1 and SDN2 can be taken through the normal, for the trait efficacy and field performance, that can be tested through ICR ECRIP guidelines or any other extant procedure for evaluation, release, and notification as per the legislation available, which is mainly the SEED Acts and related acts, not the EPA Act. So SDN1 and SDN2 should be freed from the regulation after the initial registration with IBSC and information to RCGM. The data requirement which was suggested in this draft guidelines of DBT is not needed actually, because it is unnecessary and wastage of time and effort. So this thing has been summarized. This is given in the policy brief. One can easily download and see the large number of references also have been given. Where SDN1 and SDN2, after initial IBSC, they can go to the SEED Act channel for the trait evaluation and efficacy. Whereas the SDN3, it can follow through the normal route of RCGM GAC. But even SDN3, the regulation should be relaxed, because there are two very important points of opposition has been the presence of the selectable markers and the random integration. The opponents of GMO keep on telling that. And this particular SDN3, which is guided insertions and free from markers, it takes care of those two major actually objections. So this also should be regulated in a much lenient way. And this is the same thing SDN, as I told you. And one thing was suggested that even if you put the large piece of DNA, which will come under the SDN3 category, for example, gene replacement from another variety, or we can take from the primary gene pool.
Sir, can we be little short, sir? Yeah.
Just one
Sir,
more story there.
yeah. Yes.
So this should also be come under the SDN2 category, which will be otherwise defined under SDN3 category, because we are putting large piece of DNA. So if that DNA is from the cisgenic or from the related wild species, then it should be taken as a SDN2, not SDN3, and need not to be, it should be freed from the regulation, because it's just like the alien interrogation. Similarly, the foods derived from SDN1 and SDN2 should not be treated as genetically engineered food, because they are just like very similar to the normal mutation breeding. And then the DBT draft document has asked for a lot of data for off-target mutations, but it is unnecessary, as you know that now the Cas9 protein has been created which will not go off target, and if there is any off target, it can be eliminated by crossing. So it is unnecessary to ask for so much data. And our system should be in harmony with, I think someone has commented in the chat box that there should be global harmonization, I think Professor Bansal, and it is true, it was recommended by the committee that whatever we make the guidelines, it should be in tune with the international, so that we can have easy exchange of the products and the technology. And then the group felt actually that there is tremendous benefit of this technology, and public and the policy makers should be properly actually communicated with the benefits of this technology, so there is a broad acceptance very quickly. So in the end, there was also a point that the private and public sector should come out and invest more in this technology, and that will be only possible if there is a policy is right in the place and the regulation is right in the place, so people know that when they invest, their return will come, and then we can meet this SDG 1 and 2 goals. And this is a very potent technology to ensure that the SDG goals are made at a very fast speed. That is a much more precise way to improving the plants. Thank you so much for your listening.
Thank you, sir. Thank you, Dr. Singh. And as you rightly said in your last presentation, that both public sector and private sector has to come together. So our next panelist is from the private sector. We have Dr. Amitabh Mohanty from Corteva Agrosciences, and he is currently leading the Trait Discovery Operations Group. Dr. Mohanty has been now presently in US, but he has been here in India as member of the multiple committees and task forces, including the one on genome engineering and their applications set up by the DBT. So we request Dr. Mohanty to kindly share his views on this theme of our webinar today. Dr. Mohanty.
Thank you so much, Dr. Biva and Tix for the invitation. I think this is very timely meeting, and I'm really happy to see the distinguished panelists having a great deal of discussion on this particular technology. I would also like to thank the fellow panelists for their valuable insights. I think we have covered the technical detail, the regulatory part, all of these in a really detailed way. So for my talk, I have been requested to talk about how this technology can benefit smallholder farmers. How can we collaborate better, and also the second part is capacity building and competency building. So let me focus I'll take a few slides to discuss the points that I would like to focus today. So for example, what are our priorities? Our priorities are safety of the product. We have something called as responsible use guidelines. I'm going to talk a little bit about that, and open innovation. So safety of the products. There was a question in the chat related to what is going to happen in the future, and this similar point. So for example, the CRISPR produced plants, they are as safe as the plants found in the nature. So you heard this again and again, that in the CRISPR genome editing SDN1 and 2, we do not use DNA from another species. We are modifying the DNA in the same species. The nature of technology is such that the changes that we are making, these are very specific and targeted, and these kind of changes, interestingly, does occur in nature or can be developed through conventional breeding. So there are a lot of similarities there. What CRISPR brings in this context is the possible delivery of these traits faster and more efficiently. At Corteva, we are we test our seeds derived from CRISPR produced seeds extensively field tested to ensure the specific changes to a plant's own DNA have been made exactly the way they are intended. Also, we conduct thousands of field tests over several years to study a plant's various growth characteristics, appearance, yield, resistance to diseases and pests to confirm that these edited plants grow and perform as intended. I hope that in answer one of the questions that was put in the chat, what happens once you do the editing, field generation. It's tested very extensively, Let me move to the responsible use guidelines. CRISPR is one of the many tools that we use, to deliver value to our customers, farmers. Safety and product stewardship are foundational to our product offerings. CRISPR for we are using CRISPR for advanced plant breeding, and in this purpose, we will only work with genetic material from the target plant and crop. And I think I want to emphasize that again and again. We believe in committed to open, transparent, and timely communication about CRISPR, and I have been doing this for a long time. Whether I was part of Corteva, from very beginning, we started working in this game-changing technology, and I have delivered a number of different presentation at different fora. Corteva is committed to responsible development and application of CRISPR to ensure that consumer confidence is there, and we support, a appropriate science-based regulatory oversight, which is similar to the use for conventional breeding. Corteva intends to enable individuals and organizations that want to develop agriculture products using CRISPR through access to intellectual property, technology capabilities, infrastructure, as well as scientific expertise. And I'm going to give several examples of those. And we are committed that we are going to consider diverse viewpoint in this decision-making process for products developed through CRISPR advanced plant breeding. The third part of my priority is to discuss about the open innovation. So we are committed to share our intellectual property with public research institutions. You will see examples and academia, small as well as large commercial enterprises to solve the world's most important problem, because we believe that working alone we cannot solve all the problems. We have to work in collaboration, and we do this independently as well as in partnership with the Broad Institute, MIT and Harvard. Our IP supports advancement in trade, plant breeding, enabling technologies, biologicals, digital solution to create local solution for problems around the world. Let me cite some of the examples, so probably some of you are already aware of these examples. For example, we have a collaboration with Singh on maize lethal necrosis disease. This is a devastating disease that impacts a number of different countries in Africa. We are working with Singh to solve this problem. There has been a lot of work in this area, including training of Singh scientists at Corteva headquarters in Johnston. Similarly, closer to our country in ICRISAT, we have an ongoing collaboration with ICRISAT on a number of different areas. For example, they are in crops such as sorghum and millet. In sorghum, we are working for striga resistance, and for pearl millet on rancidity. And this includes not only the access to the technology, but also training. For example, already a couple of scientists from ICRISAT has got trained for several months in ICRISAT, in Corteva headquarters in Johnston. They have come back and they are able to replicate what they have learned there. Similarly, a few other examples, we have a collaboration with Jia Simplot for bruising and browning resistant tomato potatoes. We have an ongoing collaboration with Danforth Center. I'm happy to see Don here on cassava improvement. Similarly, with Embrapa on farming competitiveness and sustainability in Brazil. And another example would be Zikal for photosynthetic capacity and efficiency. So it is very, very important that we continue to collaborate with academic institutions, large and small industries, because we cannot solve all the problem of the world. Like we are aiming to have not only more food but healthier food in a planet-friendly way. So we got to use all the technology at our disposal, and we have to use this in a collaborative way. We would like to see a global regulatory policies for CRISPR, which are you can see that they are in different stages across the world, and I'm not going to get into it. Don gave an excellent overview of the current scenario. But one thing to be emphasized here is that some of the Latin American countries have taken the lead, and there is no reason we should be behind. We should follow suit, because the CRISPR-Cas SDN1 and 2 does not involve DNA introduced from other species. So they should not be treated any way different than conventional breeding. There are more information we can have in these two websites. With that, I'll stop this part. And focus on the second part of the discussion, that is capacity building and competency. And I think this is very, very critical for the country. I must acknowledge that DBT Genome Engineering Task Force, which now is known as the Technical Expert Committee, has been forefront of this. I joined this from the beginning, 2014, and extremely happy to see some of the project that we funded in the beginning, grant. They have already publication coming out of that. So it's really great to see that the money that has been put is being valuable. Similarly, I am very aware of similar funding system from ICR, which is also making a huge impact. So overall, I think it is important to have the capacity competency development continued. And as I said, Corteva is committed to support in all the possible ways, including technology access, training, infrastructure development, and all the all these aspects. And I would be happy to take questions when those come up. And with that, once again, I would like to thank the organizers for this opportunity to share my views with you. Thank you.
Thank you, Amitabh. Thank you for very nice perspective from the industry and willingness to share this technology with the public sector organizations. With the permission of the chair, sir, although we are just about touching the time, but we will just have one quick round of some of the questions which we had deliberated and what are coming in the chat box. Very quickly, one question each to the panelists for one round. Can I go ahead, sir? Yes. Okay. So, Professor Deepak Pental, sir, we have been getting a lot of questions for you, where people are trying to seek the guidance. Basically, you have stressed the importance of the need for novel strategies to deal with a lot of problems that the agriculture is facing. We have a large group of researchers, scientists who are in the audience, and we can see their request to, with your guidance on the type of crops and traits that should be taken up for applying this gene editing in the Indian scenario. People are highlighting things like traits important for post-harvesting, vegetatively propagated crops, etc. So your views and guidance on this factor, sir.
Well, every crop has issues. If you take rice, blast, bacterial blight, sheath blight, and we have to find out as good science students what intervention will give us the best results. If we have genes in the primary gene pool, by all means map them and go ahead and introgress them. If we do not have enough genes in the primary gene pool, then we can either, if the resistance is due to R genes, we have to mobilize the R genes. You can do it through genetic engineering and now through genetic genome editing also. If the wild type allele can be achieved by some STN2 type of intervention, so that can be done. For perennial crops, but for food crops with life cycle is very long, and the quality is very important. There, the interventions may be more difficult. With crops, it may be easier. So you have to see what do we know, what is the knowledge on that crop, what are the major pests and pathogens and diseases of that crop, and then what is the best strategy to use. But you need some freedom to pick and choose the best sort of strategy for dealing with a particular problem of the plant. And until unless we encourage you by giving you a field where your innovations can stick, then it becomes very difficult. For example, we were very good in plant tissue culture, and because we were averse to use genetic engineering technologies, we have become de-skilled in that. Very few protocols are available in routine matter. Now, you need to build upon the already existing knowledge. If genetic transformation technique is available, it could be used for gene editing, it can be used for genetic engineering. But if you lose that technology, and if you lose competency in that technology of regenerating plants in vitro, then there is big trouble. So at the national level, we should try to see that young people get some baseline technologies available in different institutions. Like if you clone some gene, or if you want to modify some gene, is a genetic transformation protocol available somewhere or not? If you start rediscovering the genetic transformation protocol, then you will be wasting 2 years of your time on that rather than on genetic engineering. So at the national policy level, this has to be, we have to develop facilities for genetic transformation so that your constructs can be used very quickly, and you work on your chosen area rather than being stuck on having every technique in your laboratory. That's number one. Number two is that I want to mention it to Dr. Paroda also. You know, the ACRIP system is a very interesting, very good development, and we are very proud of it. But I think it's lost its way. The ACRIP, as it is a trial system, is based at the moment on competition. You create a variety, I create a variety, third person creates a variety, and then we compete. Who is the most clever in terms of selection in the field, and who has produced the best varieties. Well, we have come to a dead end there. What we need now is different objectives in a crop of different diseases, different abiotic stresses. We work on each one of them, and then we stack it together. So the new technologies require a huge amount of cooperation and a different mindset. So ACRIP should be made an independent analyzing things where private sector and public sector both can put their varieties, and there's no conflict of interest. So these are the 2 points I wanted to make. That will help the younger people also to compete and have their individual research project contributing to the national goal of improving a crop.
Thank you, sir. Thank you very much. May I request now the chair to kindly take over and, maybe take the take-home messages from each of the speakers, sir? Would you like to?
Well, thank you very much. I think we have exceeded our time limit, and maybe many of participants have other engagements. What I would suggest is that among the panelists and the speakers, if they could give any take-home message in terms of how to move forward. What is the way forward? In fact, we know this is a good innovation, and it is relevant, it is important, it needs to be scaled. But then, in your view, what is that in priority that we must try to address so that we are able to make an impact in terms of what is already available. So could I start with the same sequence of presentations of our speakers? Maybe Dr. Vishwanath, could you, and maybe 1 minute or 2 minutes maximum for home take message in terms of what are actionable points or way forward?
Thank you very much, sir. From the overall discussion, we understand that genome editing is similar to that of the mutation breeding, only with the advancement that it is more precise and we can create a mutation in a more defined way. And the mutation created by the CRISPR technology, especially the SDN1 and SDN2, are indistinguishable from that of the conventional mutagenesis. And these mutation methods have wide applicability in agriculture. Hence, we should quickly formulate our regulation, and we make the SDN1 and SDN2 free from the regulation so that India can take advantage of this technology. And also, when compared to other countries in the public system, we need to invest more, and we have to bring more and more people in the CRISPR editing so that the technology is fully used for the benefit of the country. Thank you, sir.
Thank you very much. I think David Mckenzie, with your vast exposure with regard to regulatory systems, what is your advice to Indian system in terms of moving ahead with this approach?
So I think a lot of what I would want to say has already been said. But I would just reemphasize that I think a lot of progress has been made. The guidelines that came out from DBT and the refinements that have been added by NAS and by the consultation process, I think have placed them now in a good place. I think it is time for the government to do the needful, to actually adopt the guidelines, to release the potential of Indian researchers to contribute to meeting these food and nutrition security challenges that we all face in the light of climate change and other natural and man-made disasters. So I think the recipes are there. There is a growing consensus around the world, and I think it's just time to move forward and adopt a rational approach. Thank you.
Thank you. Very helpful. So then we come to Dr. N.K. Singh. Any important home tech you have already summarized? Please.
Well, I would like to reiterate what Dr. Viswanathan and Dr. Mckenzie has told, that the most priority is that our regulation should very quickly, they should come up with this thing that SDN1 and SDN2 are not transgenic, and they should be free from the regulation. And this will set the ball rolling. And of course, they create more projects and programs along this line. So that's the first priority, is to come out with this regulation, and a lot of discussion is going on. I don't know why it is going on for endless rounds of time. So we should try and make effort that as quickly as possible, they should make this thing very clear. So we come in the green zone, from yellow zone, we come to the green zone, and start working and delivering rather than discussing endlessly. Thank you.
Good, thank you. I appreciate that. Dr. Amitabh Mohanty, I would also appreciate if you could clarify as to whether you have also entered into any MOU or any understanding with the National Agriculture Research System, like with international centers you have done, and how can the IP issue relating to this technology, which is presently with Corteva, is made available as a national public good? Is there any possibility, or there would be restrictions? And if so, then say, for example, if there is a banana through CRISPR-Cas by NABI, could it be commercialized now? We would like your clarification on this.
Sure. Thank you, sir. First of all, I'll echo all the other experts that we need to have the science-based regulatory policy, which SDN1, SDN1 and SDN2, everybody kind of agreed that those should not be regulated. That's first, and capacity and competency building should be ongoing. I would also say that we need to continue to enable and build an environment of trust and transparency for consumer acceptance. That is important. Now coming for the technology access, as I have mentioned in my presentation, we have offered the technology to small institutions, large institutions, and there has been discussion with leading institutions like ICR. There has been some discussion at DVT level as well, for possible access. So Corteva is willing and able to provide technology access to everyone who is interested in using this technology. So and as you would have seen, several of them I listed, and there are several more which is ongoing right now.
We are very appreciative of that approach adopted by Corteva in the interest of agriculture in general and for the smallholder farmers. So with that, any other comment, maybe Vibha, you would like to make at this stage?
Thank everyone. Thank all the participants. I think close to 600 participants were online throughout, although we see the number reducing now. As you said, people have other engagements. So I think, sir, if you have, if you can conclude.
Thank you. I think, as you rightly said, maybe we have to conclude quickly. And since I have been asked to chair this, it will not be a justification if some concluding remarks are not made. And in that respect, first of all, thank you all for your participation. I think it had been a useful interface. Probably we'll need this to continue. This had been a kind of interaction with the mainly the scientific fora, but what is required is to have interface with other stakeholders. Except for Corteva, we don't have other stakeholders from the policy side, from regulatory side, and from public awareness and private sector side. So we would need that kind of interaction as well. Scientifically, everyone has supported the very idea of scaling this innovation much faster and quicker, for which we must have clear need assessment at national level. What do we need for what is more important, so that we are targeted and focused and not digress in all directions. And for that, I think it would be good if effort is made jointly by some of the organizations, including NAS, TAS, also the Biotechnology Consortium, and also the Tata Institute on Science for Society, to come out with a status paper, or what you call as white paper, on opportunities and challenges to scale genome editing in plants for our food and nutrition security needs, so that we are very clear as to where we want to go and where we should lay greater focus. And that will help our policy makers, especially on research side, to invest more. And we must double our investment, if not triple. That I also see as an urgency. We need urgently a policy decision by GEAC and the Ministry of Environment with regard to clearance of the guidelines that have been submitted by Department of Biotechnology, which clearly recommend SDN1 and SDN2 to be out of regulation. And more details have been provided by Dr. Singh in that context already. And this is an urgency. They are sitting on it. I don't know why delaying it. Having had one meeting, I am told decision was taken, but then again they have opened it for the dialogue and feedback from the stakeholders, which means again a delaying process. So we need to urgently express, maybe make a strong recommendation from this webinar that there is urgency, otherwise we are losing an opportunity. We are already much behind compared to rest of the other countries and institutions. There is also a need for partnership, greater partnership with private sector, which I think is very clear. If we have to move forward and partnership with IP protection agencies or organizations like Corteva, so that there is no later on problem with regard to moving ahead to commercialize and make available the findings of our scientists in the institutions. Otherwise, we will be stuck up the same way as we are stuck up with the GM approach currently. And uh, finally, I would like to say that the confusion in the public mind, if at all likely to be there, though we know how difficult it is to overcome the public awareness issue, but we need to invest and come forward with a more aggressive approach on public awareness. For that, more effort by media, interaction with media, bringing out popular articles. Our scientists only are happy with their technical articles, but not with popular articles and not much in the news. So that aspect also has to be addressed somehow. And it was very clearly stated that we need capacity development and also institutional partnership. So that part is important. And once we know where is our priority and urgency, I would also discuss this matter with Dr. Renusuru, the Secretary DBT, and also with Dr. Mahapatra. And through their various programs, which are already in place, like BIRAC, and also by the ICR, the support to new initiatives in this area, and more support to the scientists working, and maybe why not have a mission mode program on CRISPR-Cas technology at the national level, jointly by DBT and ICR, that we had at one time, like mission program of testing and release of BT cotton, jointly by DBT and ICR, and Dr. Pentel had made a mention of coordinated program. We did use those coordinated sites for testing and for release of BT cotton. So that kind of linkage and partnership would be very critical. So I personally would like to finally say thank you all. It had been a pleasure to listen and to gain more insight into the advancements and developments globally that are relating to this. But to me as a breeder, as someone had said, I wish we had started plant breeding research now. Then when we had started, say 5 decades ago, when we didn't know how to really bring in desirable genes into our end products. So the young generation is very fortunate to take benefit of this approach. They are lucky, they must help, and there is definite need for more of crop advancement and activities and acceleration of plant breeding work. Even FAO has clearly said that plant breeding globally has declined, and there is less use of, even in the first international agrobiodiversity congress that we organized in Delhi 2 years ago. It came out very clearly that very little of wild and close relatives have been used in the breeding work over the last 2 to 3 decades than what possibly they were before. And this provides an opportunity now to go ahead, move faster. I think Subra Chakravarti has an institute which she is heading, a great role to play in that regard, and possibly gene mining should become our now major focus, using desirable genes, and there are many desirable genes available in the agrobiodiversity that we have, which can counteract all these challenges that we have before us, whether nutrition, climate change, diseases, pests, drought, tolerance, and whatnot. I have no doubt about it. And only thing is, how can we make use of this disruptive innovation? I consider this as disruptive innovation, like, we had cell phone technology available to everyone, rich and the poor. Here it will be a disruptive technology to all breeders, whether in the advanced countries or in the developing countries. Only thing is, we have to make this technology uh, international public good, and that is the challenge. And that's where public sector has to come forward more aggressively with research findings and with the new innovations and approaches, so that we are not struck up with the issues of intellectual property, particularly when it is an issue of global food security and removing of hunger, being SDG goals before us. So thank you very much. I think we have taken more of your time, but certainly I would like to compliment BCIL and also the Tata Institute for having taken this initiative. Let's make sure that this technology reaches to end user as quickly as possible. Thank you very much.